All right, y'all ready? Okay, so last time we talked about the ribosome, and its job was to take the messenger RNA and transfer it into a sequence of amino acids. I gotta go get a pen. We want to take DNA to a protein. What are the bases for DNA? Guanine again? Oh, okay. All right, so it says if we had DNA is called a what? Structure-wise. It's a double helix, so we know that we'd have the complementary bases here, which would be what? Adenine, thymine, guanine, cytosine. So you get the picture. So we know that in order to make our messenger RNA, what's that process called? Good. Transcription. We need to use some polymerase and unzip this guy and use complementary base pairing in order to make messenger RNA. Well, what's going to be different about messenger RNA? So what? One strand only. And then what else? Good. Instead of who? Okay. So we're going to have adenine, then what? Uracil, then who? Guanine, cytosine, guanine, the bad coding, Fifi. But anyway, go on. Uracil, adenine, cytosine, and guanine. All right. So, in your books, turn to page 198. Turn to page 198. All right, everybody on 198? All right. So it says, once we've got our strand of DNA, it says we transcribe DNA into messenger RNA. So then the ribosome's job is to translate messenger RNA into a sequence of amino acids. Now, it says this is an organelle that's associated with the endoplasmic reticulum. So if we're outside of the nucleus, because the membrane of the endoplasmic reticulum basically helps to form this membrane because it kind of wraps itself around in order to form this convoluted membrane system of tubules and sacs, okay? And it says, well, some of those remember, what was the dark stain region? The nucleolus. What's its job? You said make those ribosomal subunits, right? And they attach themselves, some of them, to the endoplasmic reticulum. Yes. So if you have rough endoplasmic reticulum, we know it's rough. Why? Because it has these ribosomes attached, okay? And then it says, well, we had DNA. Remember that DNA in here? It says it unwound. We made a strand of messenger RNA by complementary base pairing when we broke the hydrogen bonds. And it says this guy will also travel out through a pore, and it's gonna be fed through that ribosome so that we can end up with an amino acid strand linear sequence. Now, let me say this real quick. All of the ribosomes don't attach to the ER. We do have some free-floating ribosomes out here. Yes?
My question was, is the subunits, once they get put together out there, their job is just like to read the program that the messenger RNA is given it?
Right.
And it puts together based on what it's reading
the sequence. Correct. That is correct. All right, because we want to call the messenger RNA, well, we say DNA is a genetic code, and then we say RNA molecules are the translators of the code. Now, the first RNA molecule that we looked at was the R, ribosomal, because this is the ribosome, so this is ribosomal RNA. And then we have over here some what? Messenger RNA as well. And then the guy that's going to help us, so in other words, here's our ribosome. We didn't give it a color since it's red up there. So here's my ribosome. Let me take a part of that out. So here's my ribosome. Here's the other part of my ribosome. And then also floating out here we have some tRNAs. Just put some on. Uh-oh. All right, so we have these guys that are floating around out here, and these are called your tRNAs, or transfer RNAs, because what their job is to bring over some amino acid, okay, and complementary base pair with this sequence. Okay, so if we were looking at complementary base pairing, what's going to pair to this code? So now let's say, so we read every 3 bases, and every 3 bases, that triplet, so every 3 nitrogenous bases, we want to call that a codon. And then what the transfer RNA has is the what? The anti-codon. Okay? This, an amino acid. Okay, so what we're going to do is use that chart to see what protein it's saying that we're going to make. And hopefully I didn't put any stops in or whatever. But all right, so it says using complementary base pairing, this is the actual code, AUG, that came from DNA. So the little handy glove comes in, and he's going to have what code? What's the anticodon that's going to be there? Good, UAC. And then you're going to look in your chart, and it says UAC has what? Well, not UAC. You have to use this because it's complementary to the codon. This is the codon, so you have to find AUG. What does AUG stand for? No, ma'am. Look in your book. It's good. Methionine. Everybody see methionine? Yes? Yes? Look at AUG. Everybody see AUG? Got one still looking. You find it yet? It's in the first row. First column, excuse me. Row, column, whatever the peanut butter. Oh, and it's in green. You got it now finally? All right. So we said that that's going to be methionine. And methionine is a what? An amino acid. So then this ribosome is going to shift down and read the next three bases. We know that the messenger RNA has the complementary base pair. So what's this messenger RNA going to have on it? G, G, C. Okay, but we're going to look in the chart for this CCG. What do you find? Proline. Everybody get proline? Just make sure everybody's seeing the same thing. Yes? No? Proline. Everybody see proline? PRO? So then that's going to be the next amino acid. So what do we have to do now? If we're building a linear sequence, what do we need to do? What do we have to stick in here? Between here and there, what do we have to have? Somebody said it. Where's it coming from? Absolutely. He said, we're going to build a peptide bond. So here's our peptide bond. So it says we're going to take away what? Two things in order to make a peptide bond. Say what? What? Good, a hydroxyl and a what? And a hydrogen. All right, this guy's going to slip all the way down here and he's going to read these three. What's on the glove? On my transfer, oh, it said A, U, G. So we know we got another who in here. So it says UAC is what? Tyrosine. TYR. Okay, so what are we sticking in here? Peptide bond. And this will go on. See, this says, somebody read, what else did it say about this guy? The start. So that tells you to begin the process. And usually there's also a stop codon to let us know that we're finished making the protein. Cool beans, huh? Help us. All right. You need to be taking some notes. Remember our discussion. All right, here we go. We are done with that over there. Okay. I'm going to go back to this slide, and I'm going to try to draw it in on this slide right quick. Yes. Yes.
So what determines where they start and where they stop?
The codon.
The codon itself.
There are start ones and there are stop ones. Okay. He don't want to work today. Ain't nothing working today. What's up? Yes.
When you put up the DNA line and then the messenger RNA line, we knew that the four differences at the top was the uracil was the only one that wasn't in the DNA. How did we come up with the other different?
Down here?
Yes.
Oh, I just threw some out.
Oh, there's no particular sequence they.
Yeah. No, I mean, you'll be given whatever on the test.
Oh, okay.
Oh, you'll have a strip and you'll.
Okay, so then when you drew the glove, if you will, the messenger thing,
No, not a mess. Well, the transfer.
the transfer.
Mhm.
Put that in and came up with the GGC. How do you determine what that is?
Where's the, you mean this?
Yes.
Because it has a complementary base pair to that. Cytosine always pairs to guanine. Uracil and adenine always pair together. Or,
That's what we should know then.
correct.
Okay.
hmm Yes, guanine always pairs, so complementary base pairing, if we're looking at the RNAs, guanine always pairs to cytosine, and we know uracil always pairs to who? Adenine. Okay? With RNA. But if we were doing DNA, we know that thymine always pairs to adenine. Say it again.
Guanine with cytosine, adenine, uracil.
Correct. In the RNAs.
In RNA, right?
Right. Yes.
It shows the spellings, it shows it lined up, which pairs.
Yes. And back in chapter 3, we learned that in chapter 3. And they do it right there on page 190, right at the top.
Or we have to remember these groups.
On page 198, the chart that's given, second base chart, those are facts.
Yes.
Since that they won't change it.
Correct.
This is what it's gonna
Correct.
be. Okay, so then, what we should know is what the pairing groups are.
What you should know is how to pair them up. Because you're going to get a chart. You don't have to memorize anything out of the chart. So you will be provided with a chart, and you'll be provided with DNA. You'll need to make your messenger RNA and your protein. Yes.
I know we're gonna have to,
No. You just need to know how to use it.
I
Yes.
know we're gonna have to know the 4 base pairs as well as uracil and all of that, but are we gonna have to know like the
Yes.
RNA, what it is,
Yes.
like the 3 subunits?
I mean, you're going to figure that out from this.
We'll have this chart.
That's what I just said.
Okay.
Yes. Hey, I'm hearing too much talking and I can't answer these questions. So if y'all need to carry on the side conversation, just whisper. All right, yes, sir.
Will we draw anything where we have to take out each should? Because there's a
No. No, you already know how to do that, or you should. No more disaccharides and polysaccharides, and so you should already know how to do that because you learned that before. Now, for those people who are taking a massive final at the end, then, I mean, you know, you might see it again. Yes, sir. You mean this thing? It's a ribosome. It's 2 parts.
And the 2 parts are between DNA and RNA.
Well, it's just, the only reason why I'm showing it here is because this is that guy basically over here that's attached where the messenger RNA is going to be fed. You might not see the DNA. We've done because where do we make the messenger RNA? Where do we do that? We were here, right? In the nucleus. So we won't be with DNA. This guy's going to come out through one of those pores and go so it can be translated. Cool beans? But I did it beside DNA. Why? Because, right. Everybody good? All right, let's go. Didn't we do that the last time? All right, here we go. Endoplasmic reticulum. All right, in lab today, somebody was asking, Savannah, what are these? Did you ever figure it out? So what does it say at the top? Can you see what's at the top? Yeah, can you see my little arrow? It's yes. All right, so we got endoplasmic reticulum. Cool beans. So those of you who didn't label your stuff in lab, remember we said the endoplasmic reticulum just a minute ago was associated with what? The nucleus. Because we said the membrane of the endoplasmic reticulum helps to form that outer membrane that we call the nuclear membrane. Yes.
All of those tubes that are extending out.
All of it. All of it. All of it. All of it. And this is just a small portion of it. Yes, sir.
The tube would be the smooth and the other would be the rough.
That is absolutely correct. All right, one of the things about this, look at this membrane, because it's very important. How many people, and I can tell none of you, okay? But I'm just saying, get the help that you need so that we can succeed. Okay. Okay. So, anyway, okay, let's look at this guy. Let's look at him. All right. You can see that he has this double membrane structure. So here's membrane, here's membrane. But one of the things that you want to notice about this guy, especially when we start looking at the Golgi, all of it's connected. Every last bit of it, okay? These are not separate sacs, okay? Every last bit of it is connected with the other part in some form or fashion, okay? This part that you see in the middle, the red part, that's going to be the lumen. And then we see again that someone pointed out that there was smooth ER because smooth ER did not have the ribosomes on it. And then this stuff that's all studded, that's going to be your ER with the ribosomes. We want to call it rough ER. All right, here we go. So we said it's bilayer membrane. And then we said it has a lumen because it says once we take this messenger RNA and send it through the ribosome, it says we're going to be reeling off a protein. And it says that protein is going to end up, so here are my different amino acids attached by these bonds. It says it's going to be shipped into the lumen of the endoplasmic reticulum. The lumen, the red or pink part. That's a lumen, yes, sir.
Is that like the inside membrane or is that filled with like a...
It's like a solution in there, okay? And then that's where the proteins will be shipped to, yes, sir.
What exactly is the function?
Okay, well, it's a manufacturing site. And so it's manufacturing who? The rough part is manufacturing what? No, the proteins. So it's a manufacturing site, and it's manufacturing these proteins, and then it's going to package them so they can be shipped about the cell. All right, so it's biologically important. Why? We said it's a manufacturing site. He took it from me before I could get there. Good thinking. So it's a manufacturing site. We said there's two kinds. The smooth, that's going to do lipids and some carbohydrate molecules. We said the rough, we know that it's going to deposit the proteins. Okay? And we know we need proteins because we need proteins for receptors. We need proteins to act as enzymes. We need proteins to act as signal molecules. We need proteins to be hormones. We need proteins, okay, for everything, okay? So they have many structural molecules, okay? So they play many roles to carry things like oxygen, for example. So hemoglobin is a protein with some iron, and its job is to carry oxygen. Today we looked at red blood cells. I won't tell what they learned.
That's why it's a big deal if you have low iron, you're not getting enough oxygen.
That is kind of true. It means you're anemic. Correct. And the doctor's going to say, you need the iron, but the iron is for a particular reason. Remember, how many subunits did we say hemoglobin has from last time? How many subunits did it have? 4, 150 amino acids each. We said what was going to hold the 4 together was what? A metal, and that metal was iron. So this is the guy that's going to hold those together. And it says, because if we don't have enough iron, it says we're going to be anemic. Okay? And anemic, the word anemic means a low oxygen carrying capacity. So we need the iron, okay, so that hemoglobin will be in its active form so that it can carry those 4 different molecules of oxygen. All right, rough ER, y'all with me or y'all gone somewhere? Some of you are. All right, rough ER. All right, we said this, sometimes it says once these proteins have entered the lumen, it says they may be modified by enzymes present in the lumen. So in other words, sometimes the length of the chain or sequence needs to be clipped in order to make it active, so it'll fold up a certain way so that it's an active protein, because we know that anything that's in the linear sequence doesn't have an active site, so it may need something removed. It may even need a sugar removed or sugar added in order to get it to fold up in its 3-dimensional form, which we call what? What's that word that we use for 3-dimensional? Tertiary. Okay. Okay. And we said we know in its tertiary form, that's when it's going to have an active site. So it says sometimes we have to modify the protein a little bit in order to get it to be active. So there are enzymes that are present within the lumen that will help to modify the protein. And it says again that modification may be the addition of a carbohydrate molecule. We may have to add a disaccharide or some polysaccharide to the molecule, to the protein, in order to make it functional. And then again, we said the enzymes may remove some amino acids in order to help it fold up in its 3-dimensional shape so that it's active, takes on its active form. Okay, it says, well, once we've got these proteins in here, it says that the ER, so it's going to pinch off and form what we call vesicles, okay? And the vesicles will take these protein molecules and transport them either out to the cell membrane for exocytosis. So that's not a good color. So here's the cell membrane, and it says the ER is going to pinch off, okay? And this is going to become a vesicle. So that guy will pinch off. Okay? And it says he's going to either go to another part of the cell or he's going to go to the cell membrane. If he goes to the cell membrane, he's going to go through a process that's called exocytosis, where this vesicle will actually fuse with the membrane and only the protein will be released to the outside of the cell. And it won't be in this linear sequence form, but since I started it over there, I just kind of kept on with this thing. So it says, then the membrane, so what happens to the volume of the plasma membrane or cell membrane when exocytosis takes place? It gets bigger because this becomes a part of that. So anytime you take stuff into the cell and we make a vesicle, an intracellular vesicle to bring stuff in, we know that the volume is going to decrease. Anytime we're taking something outside through exocytosis, then we know that the volume of the cell membrane is going to increase. So this is a static process that's constantly occurring. So it says it's going to take it to the plasma membrane, and it says it's going to be secreted through a process that's called exocytosis. Yes, sir?
So if the vesicle is going to transport the protein to another organelle, it would do the same thing that did the cell wall, fuse with that wall and release the protein in it.
Correct.
What did you say happened to exocytosis?
The cell membrane gets a little bit bigger. So the cell membrane increases in volume. And then this is happening both ways on a regular basis, so basically the cell stays the same size. After everybody's taken the next test, we had some people that were out because they were sick. So on Thursday, you'll either, I will give everybody their test back so they can take a look at it and see what they missed. That's if you want it back. If not, if you don't want to see your test, raise your hand. Okay, and everybody wants to see it. All right. Yes. Endocytosis is in and exocytosis is out. No, not yet. No. All right. It says most of the things that come from the endoplasmic reticulum, they need further modification. So most of the, especially the protein molecules, they're going to be transported out to Golgi. And Golgi's going to do the processing of the protein. Now Golgi are these 3 to 20 flattened sacs. And notice, you can't see what I drew. I'm going to move this chair. But somebody tell me the difference between what you saw with the endoplasmic reticulum and what you're seeing with the Golgi. Correct. The Golgi are these separate sacs, but they are not connected to each other. So what happens is you'll have something that comes from here that pinches off, and that vesicle will fuse with the membrane, basically become a part of the membrane. The contents will be released inside. He will make a new vesicle, and then that vesicle would leave and then fuse with this membrane until it gets all the way through. Cool beans? So it hits a cis and then a trans side. So the Golgi has what's called a cis and a trans side. All right. So again, we said most everything, proteins especially, because this is a protein processing center.
It goes through changes each time it goes through a different layer?
Possibly, yes. Okay. All right. So the smooth ER, and we'll come back and talk about Golgi in a minute. Smooth ER, we said has no ribosomes, basically makes the lipids. So we know what kinds of lipids to kind of be thinking of. So phospholipids, for example, steroids, okay? So as in your hormones, cholesterol, and then it also transports down to Golgi. The main guy going to Golgi is going to be proteins, but this also modifies those other guys as well. And again, and the reason being most things are going to Golgi because Golgi is going to determine whatever the final destination is. And again, it might be at the membrane. So we know some stuff leave here and go directly, but most of it has to be modified further in Golgi. So whatever enzyme was put on, whatever carbohydrate molecule was put on in the endoplasmic reticulum probably has to be modified further down in the Golgi apparatus, okay? Or that one taken off and another one put on. And the carbohydrate molecule that was put on here may just say, hey, you need to go to Golgi, okay? All right, so this is just showing you a picture of the endoplasmic reticulum at the top, Golgi in the middle, the cell membrane or plasma membrane down here at the bottom. On this side, it's showing you some food molecule, for example, being taken into the cell. So we form this intracellular vessel by invaginating the membrane. So the membrane invaginates. It says once the membrane invaginates, it will pinch off, form this intracellular vesicle, and it says things that also come from Golgi that we'll talk about are lysosomes, and lysosomes' jobs are to break down old worn out organelles, or also to break down these food molecules into smaller, more usable parts. Talking about the lysosomes? Yeah, they're not really a trash can. They help with modification. They help with breaking bigger things down and catabolism, like reactions. They help with breaking bigger things down into smaller.
The one thing I remember from learning about cells was my teacher told us to remember Lysol with all of lysosomes.
Right. So, I mean, that's right, and that's only part of its job, okay? And then on this other side, you see something that's coming in to the membrane. So we saw over here something was digested. Over here you see something coming in. It says Golgi's job is also to unwrap every single thing that's coming in from the membrane. If it's packaged in a vesicle, it's going to unwrap whatever's coming into the membrane. All right. Yes, absolutely. Because it packages, but it also unwraps. So it does two major things. All right, so the Golgi bodies, yes.
My question is pertaining to the lab.
Okay.
Amy, I think I did most of them right. I only had 2 questions, which I want to ask in this class. But my question is, in as far as the cell structure, you know how you just have the Golgi apparatus and then you have the lysosomes next to it. In this instance, when you look at different pictures trying to find all the names, those 3 places are specific
We use
next to that, or
yes, because a lot of times what happens once Golgi has made its modifications. Golgi's job, one of the other major jobs as far as lysosomes is concerned, is to take that protein that was created and make it into a catabolic protein, some enzyme that's going to break some stuff down. And so what happens is it makes these lysosomal packages, and they remain near Golgi for the package of unwrapping and digesting things.
so, but is that's not necessarily true to all parts of the cell, though? I mean, as
Cor-
far as the nucleus is
Right,
concerned,
the nucleus, right,
but other parts may be in different
Right,
areas,
right.
not necessarily.
correct.
Okay.
All right, consists of, we said 3 to 20 flattened sacs. We want to call those sacs cisternae. Now, one of the things about this, y'all need to, for me, the easiest way to learn this would be flashcards, because there's going to be a ton of information again in these chapters. And these words like cisternae, you want to make sure you know that they belong to the Golgi apparatus, because we're going to talk about cristae as well, which belong to the mitochondria, and you want to really get these names in the right places so that you're not confused when test time gets here. Again, we said that these guys were not connected to each other. We said, what does it do? It's a processing center, and we want to say it's principally a protein processing center. So mainly modifying and sorting proteins, okay? Determining their final location. And a lot of times, again, we're going to be making glycoproteins. Glycoproteins means that we've attached some carbohydrate molecule to the protein as a way of signaling where it's going or what it's supposed to be doing. Okay, so so again, remember we said we may add the carbohydrate molecules to proteins over in the rough endoplasmic reticulum, but it says a lot of times those molecules that have been added have to be modified, so again, they're shipped down to Golgi. So then the next thing that we want to look at is, okay, so once we've made these things and modified these things, where do they go? Where do they go? We said we know that they can either go where? To the plasma membrane. We know that some other guys basically hang around and act as lysosomes which is what Val just asked about. We know the nucleus is usually in the center of the cell. She was talking about locations. Usually we know those guys are in the center of the cell, but if you remember when we look back at the adipose tissue, the cell's nucleus was where? It's pushed all the way over to the side, right, because it's also a vacuole. It's called a fat vacuole. So it stores that molecule as energy inside of the vacuole. All right, are there any questions so far? Any more? If you look online again, right underneath that folder, you're going to see stuff on the microscope, study guides, and just a whole host of, yes.
The test coming up in here, it won't have the microscope stuff on it, will it?
On the lecture test, no, but it will be on the lab test. And you'll need to know the functions. So make sure you pull that that information that's on there and make sure you learn the functions of the parts of the microscope, because that will also be on your test. All right, so again, finally we said any carbohydrate molecule that's added, we know that it can serve as some signal molecule, basically determining the final destination. Okay, where are we going? Are we going to another part of the cell, or are we going out to the membrane for exocytosis? The other thing, again, we said about carbohydrate molecules that are added to proteins, especially if they're in the membrane, they basically serve as a sugary coating, a glycocalyx, and they help us to know whether it's our cell or whether we're dealing with a foreign cell. Okay? Yes, sir. They're used in many different ways. But right, they are energy sources, but they can also be used as signaling type molecules. And so we're looking at a carbohydrate that's attached specifically to a protein. Okay? All right, it said other parts. Yes. That's what I'm saying it. You can give me time. It says other parts of the Golgi may add fatty acids. All right, now, this information is what I've been saying all along, and those are some of the functions of glycoproteins, and then also glycolipids. Same thing. Anytime you've got this coating on the outside, cell recognition molecule. One of the things that we'll do when we get to chapter 5, we'll look at the actual membrane, okay? And we're going to look at, well, based on the structure of the membrane, well, how do we actually move things from this side to that side, or how do we move things from the inside of the cell to the outside of the cell? And it's going to be based on, you know, the structure of that membrane, those proteins that are actually present within the membrane. Because if you look at a membrane, it's not just phospholipids, even though that's a major component making up the membrane, but we also have some proteins that are also embedded within the membrane and just on the surface, either on the inside or on the outside of the cell. Okay, so go in and look at your notes. All right, it says, when we leave the Golgi apparatus, it says some of the vesicles that are leaving the Golgi, it says some of them go to the plasma membrane for exocytosis. So it says whatever's inside, it says it's going to go, that vesicle is actually going to go fuse with the membrane and only release the contents. Okay? Because we know that proteins are what? Too big to fit between the phospholipids. So they have to be packaged in a vesicle in order to get outside of the membrane. It says others, it says they may travel to other organelles within the cell. It said, but then it said some of them float around in the cytoplasm, and we want to call those lysosomes. Okay, so we said some of them go fuse with the membrane, and we said that they go through a process of a secretion that's called exocytosis. So in other words, your protein hormones, when they're synthesized, it says in order to get the protein hormone into the bloodstream so it can get where, it has to be what? Sent outside of the cell. It says some of these other proteins, it says they may go to another organelle. It says, and finally, some of these guys, when they come from the Golgi, it says they just kind of float around in the cytoplasm with the proteins in the inside. It says when they do that, we want to call these lysosomes. And the word lyse means what? To split, absolutely. To split. Okay? Possibly. All right, so the lysosomes, we know that these are specialized vacuoles or vesicles, and they are filled with hydrolytic enzymes, because we know hydrolysis means to split water. So these guys are going to be involved in reactions that are breaking things that are big down into smaller parts. So we're going to be taking bigger things and we're going to be breaking them into smaller parts. So if you've got, say for instance, somebody got cut in lab today. So there's some lysosomes that are cleaning up that mess. They're cleaning up the damaged cells at the wound site. Okay, and they're also making sure that organelles that were damaged, they're cleaning all that up. Okay, so lysosomes' job is to help with that. We also have lysosomes that are inside of, say for instance, some of the white blood cells that carry on these hydrolytic activities. So once you get a bacteria in there, we don't want that guy. We want to digest that bacteria cell. We want to kill him. We want to be able to split him open. Okay? All right, so capable of hydrolysis or digesting reactions. Wow, yep. All right. So as we saw in the last slide, we said anything coming from the outside or inside the cell, it says it's going to fuse with this guy. So basically, we're going to fuse with that and make this one big guy in order to digest away. Okay, so we've got this guy fusing with something over here, and it says we want to be able to break that guy down into smaller parts. Okay, it says if we're looking at lysosomes and white blood cells, it says their job is going to help you get rid of that, disease-causing bacteria and viruses. Again, we talked about wound sites. The other thing, it says we've got these guys that are first at the wound site. Anybody know what those are called? Which ones? The neutrophils. Those are always the first one at a wound site, trying to make sure the bacteria doesn't get a chance to spread itself elsewhere. And then again, we said the other function. So we said they function as repair and defense, but they also function to do what? Digest macromolecules. Okay, so we can take these food molecules and put them into usable parts. Okay, so we said two functions, repair and defense, but also to digest some molecules. All right, we'll stop right there. And next week, you have a quiz, lab quiz over 5 and 6, because we're doing 5 and 6 in lab today. What else? Next week, if you're making note cards, you want me to look at note cards or whatever, just make sure you bring those today's Tuesday. Thursday, excuse me. So next class. But I will get a head start because this test will have chapters 4, 5, 6, and 7. So we're going to take, yes, because all that goes together. So the cell and all the cell parts are 4, 5, 6, and 7. 2 big things that we'll have is cell respiration and photosynthesis. Okay? Yes. And that's 6 and 7.
I noticed that in the diagrams that I've been looking at, the vesicles and the lysosomes and the vacuoles, they look very similar.
Yeah.
The vacuoles. How do we tell the difference?
What's in them?
Just what's in them?
Yeah. Solids for vacuoles generally, and except for